Okay, hi David, how's it going?
Doing very well.
Good. So we're now into the second of actually four casts that we're doing on the classification of viruses, and this time you want to talk about the negative strand RNA viruses.
That's right. So we spoke about the positive strands, and so positive sense RNA viruses, and so now we're going to talk about the negative sense ones. And so if you recall, when we discussed the positive strand, positive sense RNA viruses, we spoke about the fact that when they come in, they bring in the genetic material, and then it immediately interacts with ribosomes in order to be translated into the viral proteins. Well, the negative sense RNA viruses, that can't happen. And the reason is that this information doesn't encode the proteins. It is complementary to the RNA that encodes the proteins. So we immediately face a problem. This RNA is going to enter a cell, but it can't be translated into the viral proteins. So what's going to happen? I mean, how is it going to be able to be replicated? Well, they have a very clever strategy. They bring in their own polymerase with them into the cell. And once it enters into the cell, it then makes the, it then makes the positive strand using this as a template. So it'll come in and start over here using this, the purple one as a template, and make this. And it'll make a whole bunch of these. So that's the first thing that happens. So
just pause for a second and just explain. I mean, what is it about the negative strand? Which, I mean, obviously it's encoding information which can be translated into the positive strand.
It's not translated. It's called transcribed.
Transcribed, which can be transcribed into the positive strand.
Correct.
So why is there nothing in the cellular architecture of a cell which it's targeting which cannot perform that function, whereas with a positive strand there is something which can perform that function?
Right. So, well, in the positive strand, the positive strand has the information which allows it to be translated into proteins, and then a new viral protein is being made. In the case of the negative strand, it doesn't have the right information for that translation process, and furthermore, cells do not contain RNA-dependent RNA polymerases. They can make RNA, but they use as a template DNA for making that. So they, as I mentioned before, if you're thinking about DNA, it's more like a computer storage device. And so what's available to the cell is the ability to read that storage information, but it can't read they can't read the information which is coming in from the outside in a different format. And so this different format is RNA, but the virus brings in a protein that has that inherent capacity to replicate RNA into another RNA molecule. So that is create the complement. Cells don't have that activity in them.
Okay.
So what happens initially is that we have multiple positive strands made, and then they can be translated by the ribosomes, just like we saw before. They can be translated into the viral proteins. So in this case, we just had the one viral protein which was able to make these strands. Now we can make lots of new proteins, including this one here, which can take one of these and make lots more negative strands. So it's similar to that green protein that we brought in, can be used to make more negative strands. But it can also be translated into the other proteins that are necessary for the viral replication, right? Ones that are necessary for making the proteins that are necessary for assembly, potentially ones that allow it to overcome the viral, the host immune system, things like that. So that's the strategy. But once it's made lots of these, it's been used as a template to make lots of these, then we can start to assemble viruses and send them on their way.
Got it.
So that's the negative stranded RNA viruses. Once again, we want to sequester these what are called replication intermediates, where we have double-stranded RNA, because we don't want the cell to see that that's happening in the cell, because that's a tip-off that there's a viral infection, and then it will engage in various ways to try to escape from that viral infection. There's one more class of RNA viruses, which is a variation on these, and that is that the genetic material that's in the virus particle itself, when it's outside a cell, is double-stranded RNA. So it's double-stranded RNA. So it has both strands, but once again, it is sequestered. And once that enters in the cell, very interestingly, when it's replicating, it sort of stays inside a virus particle, and then it's sequestered from the cell. Cell can't see that there's double-stranded RNA in it. And what happens is that this is used as a template, so it has to bring in its own polymerase again. And so this strand here, the negative strand, is used as a template for making positive strands, and those positive strands are sent outside the particle into the cell, where they can once again interact with ribosomes to make new viral proteins. One of the viral proteins that's made, of course, is the RNA-dependent RNA polymerase. So we make more of these. And once this happens, double molecules are being made. So in this case, we have to once again bring in, this has to have the polymerase with it when it enters the cell. We have positive strands that are extruded from the subviral particle. So it's not the whole virus, it's just part of the virus. It's been partially disassembled to allow this process to occur, but it's ejected, translated, and then copied into double strands, which are packaged into the viruses.
That's very interesting, David. Perhaps in the last few minutes of this cast, it's worth just giving. Can you give some examples of the more commonly found RNA viruses, whether positive strand or negative strand or double strand?
Sure. So rhinoviruses and polioviruses, for example, are positive-stranded RNA viruses. Ebola, rabies viruses, influenza viruses are negative-stranded RNA viruses. And the double-stranded RNA viruses include the rotaviruses that cause gastrointestinal diseases. So those are representative members of those particular classes. I did want to mention one last thing, which is that when we have mostly been representing these genomes, as if it's a single RNA molecule, a single negative strand or a single positive strand or so on. And in fact, there are viruses, and the same thing with the double-stranded RNA viruses. There are, in fact, viruses that have what are called segmented genomes. What's that about? Well, if we think about human genome, we have 23 sets of chromosomes, 46 chromosomes altogether. Each one of those is a separate DNA molecule. So each one of your chromosomes is a separate DNA molecule. So you have, in fact, a segmented genome. Your genome isn't all just one molecule. It's in segments. The same thing is true from some viruses, including, for example, the influenza virus. So instead of just having one piece of RNA, they have multiple RNA molecules. And you can see that just like us, where we can have a large different variety of traits between parents and children because of the different assortment of chromosomes that we happen to get from each one of our parents, the same thing can be true with the viruses, that they can evolve, they can change more readily, because if a cell is infected with two different viruses that have these multiple segments, it could come out that the segments in the progeny virus are not representative of just one parent or another, one virus or another that infected.
As having two or more parents for each particle that's created, they're presumably supposed to be replicating to make identical copies of themselves.
Well, so it's not, they have two different parents because they have, in fact, two different origins. So when we discussed influenza viruses, we referred to them as like H1N1 or H3N2. So those have, they have a common ancestor in the distant past, but they are separate viruses now. And if those two different viruses got into a cell, what could come out could be a mixture of those different parental viruses. So it isn't, not all, and viruses are varying all the time, but even among a particular species of virus, they can have separate evolutionary tracks and then come back together to infect a particular cell. So they're not all clones of each other in any case, but there are ones that have been separated in evolutionary space and then come back together, and they can potentially engage in this process. This process is called reassortment in the case of the influenza viruses. But that's one of the things that can promote evolution, is the capacity to have these segmented genomes that allow for mix and match from different input viruses.
Okay, interesting. Thanks, David. So next time we'll discuss DNA viruses.
All right, very good.
All right, good to see you.
Thanks a lot, sure.